Hello. Hi, everybody. We are live now, and I'm sorry that we're a couple minutes late. So I can give all the time to our experts that are on the camera here. We're going to jump to that right away. We have two great speakers here. Larry will lead us through some of the what the program looks like. Craig is going to talk about the practicalities of what it looks like for a sponsor, what his priorities are, how he sees the output bringing value there. We'll save time at the end for questions. Down at the bottom of your screen, you can key questions in. With no further ado, Larry, over to you. Dr. Schwartz.
Thanks so much, and thanks for having me here virtually. I look forward to one day soon getting together in person. Can we show the first set of slides?
First slide, please. There we are.
Okay. I don't actually see them, but I have them up my own screen, so that's absolutely fine. Bill, I'm just seeing you, so that's even better. So you know, you don't hear many talks on cancer beginning with a fairy tale, and this is supposed to wake you up or actually put you to sleep. You are seeing the fairy tale slide, correct, Bill?
Yes, we are.
Okay, perfect. So this is the Princess and the Pea. Once upon a time, there was a prince who wanted to find the princess, but she had to be a real princess. So she traveled around the world to find one. And only when there was a real princess, because she had felt the pea right through 20 mattresses and 20 feather beds. So the prince took her for his wife because now he knew he had a real princess. So you could ask yourself, what does this possibly have to do with cancer care and with response assessment in oncology? And let's see if I could just advance the next slide, or you could, did that advance? I'm gonna have to call out just to advance the next slide. Yeah. So really, I want to explain very, very briefly the foundation for how we do response assessment in 2020. And that relates back to a paper that was published in 1976, where the effective measuring error on the results of therapeutic trials in advanced cancer was actually published. And the basis of this story really is the story of the Princess and the Pea. Clinical trials in oncology were just starting. We wanted to know what the bias, what the measurement error was for physical palpation. And so these two oncologists, brilliant oncologists, invited 16 of their friends back to their hotel room at ASCO, and they had placed 12 simulated tumor masses under mattresses, and they asked the oncologist to palpate the pair. And only when the pair had a difference of 50% could these simulated tumors be palpated differently. So in fact, that became the ground truth for what we even use today for response assessment. 50% bidimensionally converted to 30% unidimensionally, the so-called RECIST partial response category. Next slide. We know, however, that there's been an enormous change in the landscape of imaging and drug discovery since 1976. We have many, many new modalities with which to study. More importantly, for this project, we have many different ways of analyzing those images, be it with 3D, be it with volumetrics, be it with artificial intelligence and image post-processing. You all know far better than I the change in the landscape of drug discovery, and we'll be hearing about that in the next talk as well. So the question for 2020 really is, how can we combine the changes in imaging with the changes in drug discovery and image processing really to provide better biomarkers that you'll hear about can provide less bias, greater accuracy, but with which, most importantly, we hope will be able to decrease the size of clinical trials, decrease the cost of drug discovery, and identify more rapidly and in a shorter period of time key biologic steps and ultimately agents that can be regulatorily approved. Next slide, please. I'm going to conclude so that we really have more time for questions and answers and to hear from Craig as well. Here's the validation set of an artificial intelligence project that we did with standard of care imaging on CT scans in a cohort of patients with melanoma who were treated with immunotherapy. And what we were able to do here is better separate those in these Kaplan-Meier curves, those long-term survivors that we show in the AI signature, the 203 patients, from those patients with short-term or who did not do as well on the study. And we see how that compares to RESIST and how we could better differentiate populations using some of these artificial intelligence techniques that we're going to be exploring in this volumetric CT and automated RESIST project. My next and last slide shows what this would mean for an actual trial. Here we take two drugs, and these are older targeted therapy drugs where we've had the answers already, but we don't think that these results really will be much different with any combination therapy or IO therapy at all, where we compare the tumor kinetics and modeling holistically with a volumetric assessment, a better measure of the overall tumor burden, which we will, within the next year, hope to be able to do in a fully automated manner, or at least in a semi-automated manner, with which we could not possibly do before. And when we apply kinetic modeling to this, we see the decreased number of patients that we'd actually need to enroll in a clinical trial to obtain a similar result as when we had measured with the historical unidimensional measure. That's not to say that the unidimensional measure isn't valuable at all, but we think now combining the new technologies in drug discovery with the advances in the acquisition of the imaging, but now especially with the advances in the processing of imaging using artificial intelligence, we could do things that hitherto were not possible manually by a radiologist. So that's the essence of what we want to do, and I'll turn it over to Craig now.
Thanks, Larry. If we can just have my first title slide, just by way of just the introductory comments before we get into my slides. First, I wanted to thank Bill and Project DataSphere for inviting me to participate in this breakout session. I think it's actually very visionary and very much future looking, but really in a practical way, having applications today in the near term. And the fact that this is a subject for this breakout session is a great testament of Project DataSphere and everything they're doing to accelerate oncology drug development. If I can have the next slide, please. It should say framework. Framework for accelerating oncology drug development. So I wanted to make a few points here. First of all, that sometimes we think about these new technologies as being very interesting and very aspirational, but not necessarily applicable to what our goals are, from a sponsor point of view and accelerating oncology drug development. I want to make the point here that these are really this is really not the case in this setting. These are goals that are very achievable, and in fact, many of the sponsors, including the one that I work for, Janssen R&D, are already putting in place the internal capabilities and the individuals and the teams that are already developing internal capabilities to collaborate with key stakeholders like Larry, regulatory affairs, other external collaborators in this area, to really begin to apply these type of new radiomic technologies. I'm hearing a lot of background noise. I don't know if you hear it as well. Okay, thank you. So what I wanted to do is share with you some of the areas where we feel that in the near term, some of this technology can already be applied and developed, I should say, for application for oncology drug development. And if we look at from a prognostic point of view, developing radiomic signatures that could perhaps identify, for example, a subgroup of lung cancer patients who may be more at risk for brain metastasis. In this way, if we had a drug that perhaps had a profile that would suggest that it could be highly active in this subgroup of patients, we would want to enrich for this type of group of patients in a study where we could really evaluate the treatment effect. And again, there are signatures that Larry and his team and others are working on to be able to identify this type of patient population that may be at risk for an early metastasis to the brain. Similarly, there may be radiologic features, again, on baseline scans that could suggest that the patient's tumor has a certain genetic subtype. Again, we are screening more and more patients with solid tumors and doing molecular profiling, but sometimes, for example, the tissue is inadequate. Sometimes the test is not available. It's not part of routine screening. And if we can start with identifying these patients through a more standard CT, MRI, radiologic signature, this would allow us to do some broader pre-screening at baseline to identify patients for targeted therapies. If we look at more of a predictive response, so developing response signatures that could predict patient benefit to certain treatments. Again, if we look at the lung cancer example, there are patients, for example, non-small cell lung cancer that we may be able to stratify early on for a higher likelihood of response, for example, based upon a radiomic signature to, for example, checkpoint inhibitors. And so we get to a setting where now maybe instead of having 25% response rate, we could in fact increase that response rate to a much higher percentage. And then again, that helps with accelerating drug development, as well as identifying new investigational agents that may be highly effective in an earlier time frame. Also, there's key clinical questions that, again, with a predictive signature may be more likely to be answered. So, for example, which patients may need a combination of a checkpoint inhibitor with a chemotherapy agent or regimen, or which that could benefit perfectly fine just with the checkpoint inhibitor. And finally, also looking at the early radiographic changes after the baseline. So, for example, when the disease assessments are done, maybe as early as week 4, week 8, can we start to see certain changes that would correlate with long-term outcomes, or prognostic changes that would suggest that the therapy is likely to be ineffective, and perhaps before overt progression, a patient could benefit from earlier intervention with more active agents in that setting. Can we go to the next slide, please? So it's my last slide. I just wanted to share with you some other aspects in which these radiographic disease assessments, when we integrate the new technologies that are being developed, can really begin to not only accelerate drug development, as I highlighted in the previous slide, but also to begin to facilitate and in many ways accelerate regulatory decision making. And the focus here is on how we can improve the radiographic disease assessments. You heard from Larry, one of the areas is around developing algorithms where we can begin to focus in, or the computer can begin to focus in on target lesions, automating their identification and their measurements. And as you know, there often are controversial target lesions or measurable lesions where, depending upon where the cursor is put, it can be a 10 millimeter lesion that qualifies for a measurable lesion, or it could be, again, based upon arbitrary placing of cursors just falling short of that threshold, which, as you know, is a RECIST criteria at baseline for measurable lesion. So again, if we can increase the accuracy with some of these algorithms that begin to automate these type of measurements, again, we can really improve on determining the treatment effect and be able to have robust disease assessments, more robust than are available today. Sometimes we need to use more sensitive modalities. In this case, I provide the example of the PSMA PET, where again, we know this is a more sensitive modality to detect metastases in prostate cancer. If we are developing adjuvant therapies, it would be, again, a technology and modality by which metastases can be counted as events using this technology, which is, again, on the verge. There's some tracers that are on the verge of being approved, and this could be incorporated into acceleration of adjuvant development programs in prostate cancer and be able to at least complete them with earlier identification of events and shortening the timelines to regulatory approval. As I mentioned, there are some initiatives underway to try to automate the RECIST criteria. Again, this perhaps can not only increase accuracy of the measurements, but can also perhaps at some point replace some of the independent review, blinded independent radiologic review. That, as you know, is a requirement for several studies, several single-arm studies, where response rate is the basis for accelerated approval, and there's always a concern about bias in those measurements, in those response assessments, and if it can be done in an automated way without necessarily bringing in investigator measurements, then that becomes another way to independently confirm response without the variability that often occurs by bringing in a group of outside radiologists to do something similar. We're also looking, again, in collaboration with others who have expertise in this area to try to develop better disease assessments that could potentially predict clinical benefit in a more reliable way. And this one approach is to use volumetric changes instead of changes in the sum of diameters or the longest diameter of tumors. And again, this is something that's more long-term, but again, could potentially in the next version of RECIST be incorporated and could perhaps be a better measure, more accurate measure of treatment effect and ultimately, clinical benefit. Obviously, all of these require access to data, and I'm very happy to see that along with Project DataSphere, there are many other organizations that are working to provide data, many other pharma companies to make their data accessible so that we can begin in this type of broad collaboration with FDA and other key collaborators and key stakeholders to develop the needed validation strategies to bring this forward as part of a robust oncology drug development process. So I'll stop there, and I guess we'll open it up for any questions. Thank you.
Thanks, Larry. Thanks, Craig. I think you laid it out there. And of course, we're going to run out of time before we get to many other questions, but we will respond to all of them online after the meeting. The first one, and I think you're both going to have a comment on this. I'd ask Larry you to comment first, and that is, how do you foresee FDA's buy-in to an AI-driven solution to RECIST 1.1?
So I think initially this is going to be a tool that helps the radiologist. So it'll be, kind of a secondary endpoint. And, just like we're using AI or CAD, computer-aided detection, diagnosis in breast imaging, in other areas, we'll be able to use this as well. I think what's going to be more difficult is potentially when we look at some of these AI changes that we see in some of the features, et cetera, and we gain approval for that. Again, I think that this is going to have to be done kind of in a stepwise fashion, initially as a secondary endpoint, but ultimately I do believe it is robust and it will pan out to be able to give better information earlier.
Craig?
So I'm always the voice of optimism, and again, if we look at other new technologies and new modalities like real-world evidence, for example, we see a wonderful willingness and enforcement from the FDA to develop robust policies and guidance around use of that to facilitate regulatory decision making. So I truly believe that with the right validation approaches and with close collaboration with the FDA, that many of these hurdles can be overcome, and we could end up with the type of new modality for assessing treatment effect and for proving clinical benefit as part of a regulatory approval process.
Very good. The second question coming in from participants was, there seem to be many technology companies, including the big ones like Google and many startups, working on recognition of tumors. Are we doing something redundant here? Has this already been solved, or is somebody else working on this?
So I could assure you this hasn't been solved yet. I wish it has been. I think that we're looking at it somewhat differently than I know than many of the big players. This is actually, in terms of imaging, a relatively small space. I think for drug discovery, it's enormous and important. But what we're doing, looking at serial change on standard of care imaging and assessing that over time, both from a volumetric standpoint as well as a change analysis standpoint, is relatively novel. Remember, despite how large our clinical trials are, compared to what others speak about, this is actually not big data. So we actually have to employ other techniques in the AI realm, while others are capable of, they seem to be focusing much more on their considerations for big data.
Greg, any comments on that one?
Just to add that, we certainly need that the computer powerhouse technology that those companies offer, and the capabilities of software engineers to help us develop those algorithms. But as I look back at sort of what we've learned in the real world evidence experience in the last few years, that's clearly not enough. For all of these, when we're curating data, we do really need an oncology focus and disease expertise to be able to really evaluate the completeness of the data, the origin of the data, and that always requires more of a disease expert in the radiology and oncology realm to really be able to make sure that the data sets are actually relevant to the problem we're trying to solve.
Okay, thank you. Here's another one. Why is it so difficult to get annotated imaging data sets, both EHR and clinical trials, for such research?
I'll start. Unfortunately, we have only a few minutes, and I could talk about this, for hours. Pharma has been unwilling, or at least portions of pharma have been unwilling to give up, the data, even under, even under, controlled environment, and with, proper, authorization to use that. And it's been frustrating, quite frankly. The annotation is just a matter of elbow power and mouse power, to do that. So I'm less concerned about the annotation. I'm more concerned, as you are, about getting the data. It would be, this process would be much easier if we could get better, faster access to that imaging data. And I think that we could treat it both confidentially, and we could treat it in a manner that would be safe to all of pharma.
Thank you, Larry. Craig?
Yeah, so again, I can't speak for other sponsored companies, but I can say, for Janssen, we have a mandate, a corporate mandate through our CMO to actually make these patient data sets available after publication and regulatory submission. We've done this through a number of forums. We have Project Yoda, which is a collaboration with Yale, where anyone can come and make a proposal requesting a data set. And if it's scientifically sound, we provide the identified data. So again, I think that we and other sponsors now recognize that it can't be done without access to data, and we will do everything at our end possible prioritize our delivery of this type of activities that, as we said, have a real potential for helping not just our own development programs, but patients and all stakeholders, for oncology development.
If I could add to that, Craig and Janssen have been very proactive and, renaissance in this approach. And it's actually, we could see that it's actually helped already. If you look at what the prostate cancer community and Prostate Cancer Working Group 2 and now 3 and others are doing in that space, they're frankly more advanced than many. And that was due in part, to the team that was doing that, but also the willingness of several of the companies to provide data.
Yeah.
Thank you for that. And
so
that's proven out.
I think it's, we have to end, and it's great to end on that hopeful note that it is getting better. There are some companies that are more willing to talk about this. And Larry, the work you've done is now starting to give people more excitement or confidence that this is going to happen, it's doable. We're going to contribute to better, faster drug development through this program. So we will get more data. So thanks, everybody. Like I said, we'll answer the questions online. And I guess I just give you a virtual applause. Thank you.
Thank you.
Thanks a lot.